Professor Massimo Milan. I'm looking forward to hear your talk on how we can actually incorporate transcriptomic data into the weight of evidence approach for environmental risk assessment. I suspect it's going to be a bit heavy on mathematics, but I may be wrong. The floor is yours.
Yeah, thank you.
Well, if you want to reshare your
Yeah,
presentation, because we
yeah,
saw it briefly, but I think now it's gone.
yeah. Good afternoon, everybody, and thank you to invite me to this workshop. I'm sorry, I'm in other room. The connection is not so good, so I'm going to switch off the camera just for the presentation. We'll come back at the end.
We can't see it. You're not sharing at the moment.
Yeah, I know. I'm
Okay.
going to share. Yeah, it's okay. It's okay now.
Yes, it is. If you
Okay.
can go in presenters mode, perhaps perfect.
Thank you very much. So, transcriptomic is being applied more and more frequently to define the responses, the effect to toxicant in vitro or in vivo by comparing different dose level, concentration, duration of exposure, and tissue type. So, the advances in high throughput sequencing like RNA-seq enable more comprehensive and sensitive transcriptome profiling, and this allows for a better understanding of gene expression changes even at a low level. In addition, to my opinion, I know that there have been a couple of presentations yesterday on single-cell transcriptomic. To my opinion, these techniques will represent a new frontier also in ecotoxicology, being able to provide insight into cell-specific responses to toxicant, uncovering cellular heterogeneity that is missed by bulk sequencing. So anyway, toxicogenomics can provide transcriptomics, sorry, can provide very important information to define mode of action, and also for hazard prediction for environmental risk assessment, and so possibly to enhance regulatory decision making. However, the application of toxicogenomic in environmental risk assessment has been slower than expected. And transcriptomic is generally applied qualitatively in environmental risk assessment, and despite its promising potential, the application has faced several challenges. Among them, first, the genomics and bioinformatics are not yet as well established in ecotoxicology as in other fields. And this has led to a slower adoption of these tools in environmental risk assessment. Second, there is still a lack of consolidated and standardized method from lab protocol, from RNA extraction to bioinformatic pipeline. And third, there is still we face a significant knowledge deficit regarding the genome, epigenome, and molecular dynamics of many commonly used sentinel species in ecotoxicology. For example, I can mention bivalves, that are the species which are mainly considered in our study, which are crucial indicators in aquatic environment and are not as well understood at molecular level as traditional laboratory model organisms. At the same time, several studies recently propose a new model able to integrate different approach. Usually they use a quantitative wave of evidence approach, and this here I reported an example. This is a model developed by a group from the Polytechnic University of Ancona that integrate different line of evidence, here reported with different colors, so from chemical analysis, bioavailability, biomarkers, and so on, in a final index of hazard. I mentioned this model because we collaborate a lot with the research group led by Francesco Regoli, and every time when we write a paper together, we were not able to integrate transcriptomic within this model, and this is for sure a missed opportunity. And so the objective of our study, we work for a couple of years, was to develop a new methodology able to quantitatively integrate transcriptomic data in a weight of evidence approach to be applied in environmental risk assessment. So here just a slide to explain how we calibrate, validate, and test the methodology. We use more than 50 different simulated and experimental data set derived mainly in two bivalve species, Ruritapes philippinarum and Mytilus galloprovincialis, that are widely used in ecotoxicology, are two bivalve species also of high commercial interest. And so we perform the calibration by expert judgment, and then we validate our model using independent studies and in particular vertebrate species as brown trout and Xenopus tropicalis. So here I'm going to start to explain how the model works. So when we start to think about this model, we consider the hypothesis to use a list of differentially expressed genes. However, we realize that when we consider list of differentially expressed genes, we has to establish arbitrary cutoff value, like usually the fold change, p-value, or force discovery rate. And also, when we get a list of differentially expressed genes, it could be very difficult to disregard the multiple function of a single gene. So we decide to base our model on gene set enrichment analysis. Gene set enrichment analysis is a computational method used to determine whether a predefined set of genes show statistically significant differences between two biological conditions. So instead of analyzing individual genes in isolation, these analyses evaluate the entire gene set, which are group of genes sharing common biological function, representing, for example, specific molecular pathways or regulatory mechanism. So this approach had to capture subtle but coordinated changes in gene expression, and is widely used in transcriptomics to interpret those large-scale gene expression data or to link molecular changes to biological processes or disease mechanisms. So how does it work, gene set enrichment analysis? I'll go fast on this point. Gene set enrichment analysis rank gene. The first step is a ranking of gene based on a metric, usually the fold change. So the genes are ranked basing on value of fold change. Highly upregulated genes appear at one end of the ranked list, while highly downregulated genes appear at the other end. So for example, here we have the PPAR signaling pathway, we have the whole transcriptome that is reported in this bar from the red color to purple, and the black bar represent the genes that are included in the PPAR signaling pathway. So in this case, we have most of the genes that are placed at the beginning of in the left part of the transcriptome. That means that this pathway, so PPAR signaling pathway, is up regulated. So while in this case it's the opposite, we have another pathway, the KEGG cell cycle pathway, with most of the genes that are placed in the right part. So in this case, the pathways will be significantly down regulated. So is gene set enrichment analysis is able to consider all information without a priori definition of differentially expressed gene. And gene set enrichment analysis is also able to provide an important index, that is the enrichment score, that represents the degree, is a scale from -1 to 1, to which a gene set is over or under represented. And this, to our opinion, can be very useful to translate the significant pathways identified into a quantitative hazard index. So when we perform gene set enrichment analysis, we have also to define, to identify an appropriate collection of gene set. There are many different data set from, think about an example, our gene ontology or KEGG pathways and many other, reactome and many others. And in our case, we decide to base our analysis on the hallmark gene set collection. This is a curated collection of 50 gene sets that summarize and represent specific, well-defined biological state or processes. And are that we decide to focus on this gene set collection because most of single gene in this collection account to single function, so single gene set, and this avoid possible redundancy and overrepresentation of some biological responses. So the hallmark genes, the collection of hallmark gene set is composed by 50 different gene set that are assigned to one, each of the gene set is assigned to one of 8 biological categories that are proliferation, immune, development, cellular component, DNA damage, metabolism, stress response, and signaling. So when we start to develop our model, considering that we were working on considering a bivalve species, so Rhodapes philippinarum mainly, and the Mediterranean mussel, we decided to refine the hallmark gene set. In particular, we merge overlapping gene set that are gene set that share more than 50% of the genes that are interferon gamma response and interferon alpha responses, and the same for estrogen response. And in addition, we eliminate 4 gene sets that were never described in investigated taxa. And so the final set of gene set was represented by a total of 44 gene set that were included in one of the 8 major biological categories. Then we performed some analysis to verify how many genes of our target species were represented within this gene set, and we obtained that the clam and mussel transcriptome share an average of 50% of gene with human hallmark gene set. So here, the first step to develop our model. So the aim was to obtain an index able to reveal, to reflect the relevance of each biological category in terms of physiological reaction, but also the degree of its differential regulation. So this is the starting, these were the most important aim in the step that we were trying to develop our model. So the first step was the assignment of different weight, so from 1 to 3, to each biological category, basing on the relevance of the biological endpoint. For example, we assign a weight equal to 1 for the biological category for which changes at transcriptional level are less evidently associated to the onset of adverse effect at higher biological level, while we assign a weight equal to 3 to alteration related to cell cycle regulation, cell death, and cancer, and so that are represented within the biological category proliferation. So this is the final picture of our the weight assigned to each of the 8 major biological categories with a weight from 1 to 3. Then the second step was to define an equation able to consider the weight assigned to each biological categories, but also the score, so that able to characterize, able to consider also the changes occurring at the transcriptional level in each gene set. So here, this is the equation, that is the pillar of our model. Here we have the transcriptional effect for each biological categories that is obtained by dividing the weight assigned to the category divided by the maximum weight, that is equal to 3, multiplied by the enrichment score, the sum of enrichment score obtained for significant gene set within the category divided by the maximum possible enrichment score multiplied by 100. So to explain a little bit with an example, this equation, in case of the biological category metabolism, for example, we have 6 gene sets that are included in this category. So the maximum enrichment score that we can obtain will be 6. So for this pathway for which we assign a weight equal to 1.5, in case, for example, if we apply the gene set, if we perform the gene set enrichment analysis, identify just 2 significant gene set, bile acid and glycolysis, with an enrichment score corresponding to 0.5 for bile acid and 0.6 for glycolysis, the total enrichment score will be 1.1 for the biological category metabolism. So at this point, with this value, we can calculate the final transcriptional effect for this category. That will be equal to the 1.5 divided, 1.5, that is the weight assigned to this category, divided 3, that is the maximum possible weight, multiplied by 1.1, that is the enrichment score obtained following gene set enrichment analysis, divided 6, that is the maximal attainable enrichment score, multiplied by 100. So the final value will be 9.1%. The transcriptomic effect, we apply this equation for each category, can vary from 0 to 100%. So the first step consists in assign each transcriptional effect observed to one of 5 classes of effect. So we define, following many simulations, this different threshold. So an absent effect with 0% of effect, clearly, slight from 0 to 7, moderate from 7 to 25%, major from 25 to 40, and severe from 40 to 100%. So for example, in the previous example for metabolism, with a result equal to 9.1%, we consider a moderate effect for this biological category. And last step, calculate the transcriptomic hazard index. That is the final score that will allow to define to integrate all the biological categories to define the final risk. Transcriptomic hazard index is calculated based on the percentage of categories included in each of effect, as described in this equation, that calculate the percentage of categories that obtain a risk equal to absent, slight, moderate, major, or severe, each multiplied by a specific score that is here represented. So thanks to this equation, we arrive to define this index. So to summarize the main step, so we perform gene set enrichment analysis just considering the all-marked gene set or the refined all-marked gene set. We consider just the gene set with a full discovery rate, lower than 0.2. Then we calculate the class of effect for each category by applying the first equation explained before. And so we assign a risk to each biological category. And then the last step is the calculation of transcriptomic hazard index, and by integrating the result obtained for all the biological categories. So this is an example with all the biological categories. For example, we can obtain moderate effect for signaling and metabolism, major effect for stress response, slight for immune. With this output, we input all these results here, and we obtain a final index. So then we calculate, to validate the model, we calculate this index on 31 different experimental data set. So for example, here we have exposure of Manila clam to different PFAS, or of Mediterranean mussel to acidification of carbamazepine, or the combination of both, obtaining for each study a specific hazard. And then we validated this model with the external data set from brown trout and Xenopus tropicalis, and then we try to compare our index with the result described in different paper, and we observed that our index was able to summarize the main finding discussed by different authors, and was able also to complement the functional information by capturing the severity of the perturbation. In addition, we also provide 10 different data sets from slight to severe risk to a panel of 11 external experts, asking to define a risk from absent to severe. And we perform a correlation between our model output and the judgment of this panel of experts, and we obtain a very good correlation. So the index proved to be a good indicator across the entire spectrum of hazard classes. So to conclude, for sure, transcriptomic can be considered a super biomarker for environmental risk assessment, which are the most important characteristic of our model, is able to summarize large, complex transcriptomic data into a single index. We demonstrated transferability. We can apply the model to different species and also model species for sure. Is an approach very flexible, so we can customize gene set for different organisms. Is consistent with independent expert opinion and can be integrated into a quantitative weight of evidence. I will show you later the first study in which we included our index. We integrate our index with the index derived from other line of evidence. However, we are aware that we are just the first step to develop a very good model. We note that the transcriptomic still lack a quality standard and still lack of a standardized method for data processing. We need to improve gene annotation and genomic knowledge of non-model species, and also will be crucial to define a species-specific gene set. So derived from, because in this case, for example, we're using the hallmark, we use a gene set that are derived from phylogenetically distant model species. So in the case hallmark, we are considering a human gene set that are built considering human transcriptome. And in addition, the weight assigned to different biological processes are arbitrary. And anyway, this can be revised based on new evidence or species-specific peculiarities. So the model is anyway very flexible, and we can change different parameters and thresholds. And here, I would just mention this study. That was the first study in which our index has been incorporated with other line of evidence to define the quality of different farming sites of Manila clama in the Venice Lagoon. So in this case, we integrate data obtained from chemistry, bioaccumulation, biomarker, and transcriptomic, obtaining a final index that integrate all these different analyses. Thank you, and I finish here. Let's come back with my...
Brilliant. Thank you so much, Massimo.